Okay, I'll start with the introduction and then hand it over to Dora. So it's great to have Dora Hermes here. Dora works at the Department of Physiology and Biomedical Engineering at the Mayo Clinic. You just heard a lightning talk from Harvey describing a little bit of the kind of work that her group does. And Dora, throughout her entire research career, she's the research that she's done has kind of spun off these tools and ideas that have been useful to other people as well, starting from software that she developed as, I think, a PhD student, and including this work that she'll talk about today, which extended the BID structure, which was originally kind of like designed with MRI in mind, into an entirely new modality of intracranial EEG. So take it away, Dora.
Thank you so much for the introduction. I'm really excited to be here today. So I would like to, tell a little bit about our extension of the brain imaging data structure that we did, and explain a little bit of the background of why we did this, what the process is that you go through when you extend the brain imaging structure. But to first give a little bit of introduction about why we did this is why do we have standards for storing our data? And the example that I like to use is that you may want milk with your cookies. And then we know that you're going to find the milk in the fridge, and the fridge is going to be in the kitchen. And so, for example, when someone shares a data set with you and you're an MRI scientist, you want to know the repetition time, for example, or you want to know your echo time. So you want to know exactly where that lives. And for example, for electrophysiology, you want to know what the sampling frequency is. And is it going to be called sampling frequency or S rate? It would be like some people storing their milk in the fridge in the basement or on the third floor, or putting their milk not in the fridge, which would be kind of silly if we think about it. So we always want to know where to find things. And that is easy if you're maybe on your own, but even when you're, working just by yourself, you may come up with different names over time, or you may change your process, which, so you want to have as many standardized processes, especially, when you're starting to share things with more than just one person, which we're definitely aiming to do. And so the brain imaging data structure is a standardized structure for organizing many different kinds of neuroimaging data. It started out with MRI. We extended to intracranial EEG. Right before that, it was extended to MEG. It's been extended to EEG, and there's other formats in process, such as, for example, multi-electrode arrays or eye movements, or PET has just been accepted. So it's it covers a lot of different types of imaging structures. So I would like to explain a little bit more on the background of the ideas of how we structure data in BIDs. So why, if you have many different data types, for example, we collect MRI data, intracranial EEG data, why is BIDs convenient? So, and this really started with a project where we wanted to integrate multimodal data. We had we were working on an R01 between NYU, UMC Utrecht, and Stanford at the time, where we were collecting all these different data types in order to understand the underlying physiology that produces these different signals. And at the time, we were looking for a way to share that between those institutes in a way we could have adopted, like maybe the standard that one institute was using, maybe a standard that another institute was using, but it would have been, we would have had to kind of come up with our own structure. But then at that time, Chris Gorolewski was actually one floor below us at Stanford. And so I went to talk to him and see what they were up to with BIDs, because I thought maybe it only is for fMRI data. But he explained to me, this is kind of a standard structure for neuroimaging data in general. And all the metadata are human readable, machine readable, and it should fit most use cases. And it is designed to be as straightforward as possible to basically store data in a way that most labs do it. And so they had published this paper at that time where they show how, for example, from a set of DICOM sequences that you see here on the left, you can move your data into a BID structure where everything is stored within a subject, for example, separated by an anatomical folder, a functional folder for your functional MRI, diffusion weighted imaging, and then you have a list of subjects. And they chose particularly this structure because this was the most practical solution. They didn't go for maybe the best or optimal solution, but just the most practical solution that is human and machine readable and that fits the way in which most labs store their data. And the other thing that is just kind of on a high level before we dig into the details of what these metadata contain, one of the things that was very important is that they designed bids to be useful for as many people as possible. And there were a lot of the developers were involved in the structure. So people from SPM, AFNI, MRTrix, FreeSurfer, and also from the electrophysiology side, from Brainstorm, Fieldtrip, and EEG Lab, were all involved in the extension of this structure, and we're developing apps or ways to work with these data. And there are currently many online data sets available on OpenNeuro that particularly supports people to share data in bids. So it's really widely supported, and it's not only for fMRI. It's really supposed to be a community effort. And at the time when we were looking into it, it was being extended to MEG by Yomar Niso. And so Chris said, like, why don't you just extend it to intracranial EEG data? And he put me in touch with Chris Holdgraf from UC Berkeley, who was also interested in the same thing. And so Chris and I thought, like, okay, let's just see what we can do. And so we got a whole group of people together to make sure that it would fit many different data types that we collect with intracranial EEG, and that it would also be it would match, for example, the way that people would store the MEG data or the EEG data, such that it's very consistent across modality, not only within, but also across modalities. And this really emphasizes the community effort in bids, that it's a large community that's contributing. It's not just one lab that is deciding how it works. You really have to make sure that it fits what many people see. And at the same time, when we were extending it to intracranial EEG data, we had a close contact with the people with the EEG extension to make sure that it matched, and to make sure that we were all aligned between intracranial and other recordings. And current extensions also have many other data formats. So that's kind of the background of why we were starting to use bids for our data. So let me show you what it kind of looks like if you have your data stored in bids. And so the bids hierarchy for raw data. And I talk here about raw data because BIDS is supposed to store raw data rather than source data. Source data are the data that you get right off the device, such as a DICOM file, for example. But raw data are the data that you really work with in order to do your analyses. Are stored, the first level is the project. Then you have a subject at a session, which is optional, and then an acquisition. So for example, what that looks like in terms of a folder structure, you have a project, you have several subjects, you can have source data and derivatives data living there as well. You can have several sessions for a subject if you want. And then you can have the type of acquisition, for example, from anatomical maps to intracranial, PET, behavioral, or NIRS data, which is a current extension in process. And that's kind of how this folder structure is lined out that I showed you initially. You have a data set, subject, and then an acquisition in this case. And one of the things that you may notice here is that all the files are stored in with a specific name that has sub dash and then a subject label. So the standard labeling is makes it really easy for any type of processing, whether it's in MATLAB, in Python, in R, or other programming languages, to read these data, because it's always sub dash and a label, slash dash and a label, and then a particular acquisition type, and then you know what your data are called. You can come up with these names within a lab, you make the standard structures for that, and you can have optional labels for different acquisitions, for example, reconstructions, spaces that your parameters are in, etc. And so one of the first things we did is, just as a note, when I started my lab here 2 years ago, is we had, discussions on what are the subject labels, which is standard IRB protocol, but what are our sessions called? What do we call a session? What is for us a logical grouping of data according to a session? So you can decide these things for specific projects, like what works best for different projects. And that's one of the questions that we sometimes get when people start with BIDS is, what do I have to name this? This is not necessarily a session, but it's a logical grouping of data that groups together. And then of course, like what's a run? Is that one run of data, two runs of data? How do you store that? Now, the other thing in BIDS that is important is that every file is accompanied by different types of metadata, and these are specifically TSV files and JSON files. And just to show you what a JSON file looks like, it's a key value assignment where you have a, for example, the repetition time of 3 and an instruction. So its key value pairs are always stored in data in JSON files, and the nice thing is that they're human readable and machine readable. So even if you are not, if you work in a clinical environment like we do, and there are technologists who do not have a large background, they can still edit a JSON file and adjust it according to the instruction. For example, read the task instructions. That was a very important aspect for us, is that it's also human readable. And then there's tab separated file formats, which is basically an Excel file, but also a table or an array or whatever. We can use in our computational tools to read out onsets, for example, for events. When you have all your files structured that way, the nice thing is that you can use the BIDS validator to check whether all your files conform BIDS, and it will give you informative feedback on what is matching and what is not matching, whether you've missed a column, whether your columns are in the right order or not, whether you've misplaced some information, whether you have a file that doesn't form according to BIDS. And it has instructions. So it has instructions for what you should fix in order to have a properly formatted BIDS data type. So as a brief introduction, what's BIDS? So we have standard naming according to a standard naming of files, and all metadata are TSV and JSON files that are interpretable and readable. And if you, one of the questions that you may ask, like, what if I want to add additional information? What if it's not really described in BIDS what I want to do? And I want to highlight two different options for that today. The first is what I want to just add additional information, and then we'll go in how you want to extend it if you have a different structure that doesn't yet, for example, follow the BIDS guidelines. So what if you have a BIDS data set and you want to add additional event information that is not already described in just an onset and a duration and an image, which is the most simple example that we can use. And one of the things to note is that any TSV file that has, for example, event information can have additional columns, as long as they are specified in a JSON file with the same name. And one of the examples that I would like to give for how we make that machine readable is one of the things that Tal Palatia has been working on in my lab, is where we use HED, hierarchical event descriptor tags, to describe events. Because one of the things that we come across, we work with intracranial EEG data that are collected in a clinical setting. These data are recorded from subjects who are undergoing epilepsy monitoring for about a week at a time for clinical reasons to localize the seizure focus. During this period, we can sometimes record data for research purposes. We have IRB approval to ask them to participate in research. But there may be aspects of these data that we may want to annotate in a standard way. And there have been, in terms of epilepsy descriptions, for clinical descriptions, a lot of standards have been developed, but have been mostly written down in, for example, in a publication, such as you can see here. So you see these clinical event descriptors. You can, for example, call something epileptiform interictal activity, and you have these kind of names that you can call something. But one of the things that you see is that it's, there's dashes somewhere, there's a space and a dash. There's not a lot of standardization with which it is described in words in a publication. And now this particular SCORE, standardized computer-based organizing reporting for EEG, has been implemented in a standard computer system that you can purchase. But it's not freely and openly available for many scientists to use in a standardized way. So when I was when Tel started talking to me about her work that was mostly on epilepsy, we thought, like, why don't we just make sure that we can actually read this and we can actually use these types of annotations to, for example, note epileptiform activity when that happens, even if it's an artifact for our data or for other people to be able to decode or predict seizures. And what you can see here is an example of one of these, intracranial data sets where you have electrodes implanted. And these are, for example, stereo EEG electrodes where every single electrode is a probe, or surface electrodes where each channel is implemented on the brain surface, and you have a signal coming from every electrode. You may want to annotate, for example, when there is, generalized tonic-clonic, all these different types of clinical features. And what you can see here is that the table that you saw before is translated into a machine-readable set of properties where you have either background activity, interictal findings for a certain period of time with an onset and a duration, such that you can then read it out in machine-readable format. And this is what that would look like in a BIDS validated structure. If you have an onset, a duration, and then you can have hierarchical event descriptor that describes this structure. And there will be, as Otal is putting this online, making it openly available, such that you can basically choose what kind of background activity is going on. And we've already noticed that these interictal findings, whether they're low or high, are extremely useful for annotating artifacts in our data. For example, when we record data during a motor task or during a visual task, and some epochs have large epileptic activity, we can now annotate that in a standardized way in our data so that everybody knows what is in there, and it's not just saying like interictal or non-interictal or maybe noisy or whatever other people may call it. It's well described in a machine readable way. So this is one way to kind of enrich the bit structure. So if you want to add additional information, you can always add it, and sometimes even use hierarchical event descriptors to do that in a standardized way. But what if my data is not described in BIDS? For example, so that was our case initially for intracranial EEG data. There are many ongoing BIDS extensions. So I will be showing several links throughout this talk where you can, and at the end of my talk, I have a slide where I'm showing the links because many of the information I'm showing today is available online. As I mentioned, it's a community effort where we want to share data. And so if you have questions, please post them also on an issue in the different repositories. This is something that, that really benefits from a lot of community inputs. So you can see, for example, the first BIDS extension proposal was structural, structural acquisitions. And you can see here that, for example, the EEG extension proposal, these are all the extension proposals that have been now accepted in the brain imaging data structure. And you can see the intracranial EEG here. And for example, the hierarchical event descriptions that I mentioned, the EEG was accepted at the same time, ASL, PET, genetic information. So you can see that BIDS is rapidly growing to include many different, and it already has included many different formats. And these are examples from ongoing efforts. For example, connectivity schema, eye tracking, and for example, electrophysiology derivatives, where we think about how we're going to store data that are derived from the raw data sets. And so you can see here, this is one of the this is the link on the website. And if you click on these, the first step in an extension proposal is to start a Google Doc, where the structure and the content of the structure of the data and the content of the metadata is identified. And this is also how we started with the intracranial EEG extension. We started with a Google Doc where we just put a general outline, and then we shared it with many people to ask for feedback. Would this work? What would work? What wouldn't work? What matches with the EEG and the MEG description? And what we came up with in terms of the structure for intracranial EEG is this image. So you can see here that in the intracranial EEG folder, we have several files, the EEG, vMarker, vHeader, that are annotating this type of intracranial EEG data. then it's accompanied by a JSON file that stores the metadata related to like what kind of task was run, what was the amplifier that was used. Then there's an events file that has the onsets of each event, the channels, which annotates which channels were recorded. Electrodes, which has the electrode positions. Coordinate system, operative photos is optional. And then also the stimuli that were shown during this task. Participants information that has the age, the code, and sometimes the sex and the handedness of the participant. And then a data set description that also shows how you want to share these data, who should be acknowledged, what are the grant numbers that should be acknowledged, for example, all very important information. If you are a junior PI, you want to share your data and you want to make sure that it gets shared appropriately. And this is definitely question that I get from a lot of people is like, I'm going to share my data in bits. Am I just going to give it? And is everybody just going to cite it? No. Can everybody just use it? In this data set description, you can show what kind of license you're using for these data, which is very important. So you can, for example, use a CC BY non-commercial license if that's what your option is, or you can just use a CC0 license that just everybody can use it. And it depends on where you upload your data, whether it's on Open Neuro or other repositories, such as the Open Sciences Framework, where you have a little bit more freedom. But this is something that, and there's the authors of the data set that are described in this data set description. The authors of the data set are the people who curated the data. It's not just the people who designed the study, it's really the people who spent the time curating the data such that they will get acknowledged when other people may use the data and people know whom to approach for that. And then of course a general README. But I think that this is one of the data set description is really something that has, helps other people more comfortable sharing data at different stages with publications or, after they've published a few studies. And so at this kind of stage for the Google Docs stage, we faced several hurdles when we did this for intracranial EEG. And one of the big ones was the issue of IEG data formats. Because for intracranial EEG data, we didn't just have one nifty format that everybody kind of agreed was good. We had many. There's over 20 formats for EEG or intracranial EEG data. So what Chris Holdgrave did at that time is he made a Google survey where he asked people, what are you using? What's the most common format? What's the most used format? What is an open format? What is readable and writable in MATLAB, Python, R, Julia, etc.? What are the most supported formats that we can use now that are usable by as many people as possible? And so we got a long list of formats, and one of the most used formats was EDF. Now, the nice thing about EDF is that it's widely supported, and it is readable and writable already in all these different languages. But one of the main issues with EDF is that it only allows you to store 16 bits of data. The data recorded 16 bits. So larger recording sets that are common in intracranial EEG you're not going to be able to store those in EDF. So there needed to be at least one more format. And so Brain Vision was one of the formats that was going to be supported by EEG. The Python implementation had to be matched. And so that's what happened in the back end. It was a lot of, in M&E Python, they made sure that you could read and write Brain Vision format. It was already readable and writable using Fieldtrip and MATLAB. And then, so EDF and Brain Vision format were the two formats that were bits supported, which means that they're also, if support lacks, that's going to be fixed and it's fully supported in the validator and read in the validator. And then there were three other formats that are accepted. So the BIDS validator is not going to give an error if you store your data in either EEG Lab MEF3 or NWB format, but that are there for necessity. For example, for MEF3 was essential for clinical data in particular, because it has HIPAA approved encryption. And NeuroData Without Borders is one of the other formats that is supported by by groups in Seattle, by large groups in Seattle, by blanking on the name of the institute, but it's supported by a large group of people who are recording intracranial EEG data. And firing rate data in animals. And so we need to make sure that we bridge that and that we accept formats between structures. And the NeuroData Without Borders people have been also working on making sure that that is acceptable in bits. And so these formats were, after, and then after we had this set of formats, people kind of agreed that this was a good set. There are several, like MEF3 and NWB, were both allow for parallel processing because they allow you to load just snippets of data. You don't have to just load the whole data set at a time. And so these are formats that everybody agreed on were usable. And so it was actually, after this survey, it seemed to be much easier to have a couple of formats that people agreed on. One of the things that's interesting to note is that people are still developing new data formats also for intracranial EEG data. But I'm just hoping that people will converge on one rather than keep sprouting new formats, because it really makes it difficult to adapt your tools to new formats all the time. So the second issue that is just an important thing to mention is that we had to define the difference between electrodes and channels. And this is kind of a funny thing, because you sometimes call like an electrode, you implant electrodes, and then you have channels that you record. But they don't have to match one-on-one, because a recording channel in your amplifier may also record the difference between two channels. It may also be a bipolar signal. So a channel is a signal in your amplifier, whereas an electrode is the hardware contact that is implanted in one area in the brain. And so we have two distinct structures. One that is for the hardware contacts, that's called electrodes. So every contact that makes contact with the surface of the brain is an electrode, and every channel is a signal in your amplifier. Now, at the time when this definition had been quite established, and we asked for some more neurosurgeons to review this, they said, like, but actually electrodes, we call an electrode an entire probe for stereo EEG with about 8 channels on it. That's what we call an electrode. So one electrode is what we call a probe, and we call every single contact a contact. And so at that point in time, we had to defer to the fact that EEG also uses electrode as a name for a contact, or so we couldn't make everybody happy. And I think, and when we explained the structure, where we really are a community and we're really trying to do something that goes across disciplines, it's not just for stereo EEG, it's also ECOG, also EEG, also multi-unit arrays. An electrode is the same as a contact, a metal contact with the brain. But that discussion all happened in this Google Docs stage, which was really interesting to see everybody put in feedback and make sure that people are aware of these things. The other thing that happens when you make this extension is you decide what's required, what's recommended, what's optional. And that was something that's actually pretty tricky, because what is required is something that is essential to interpret the data. Recommended is very useful, and optional is something that may be useful to add. But deciding on that distinction, what was required and what was recommended, and had to match, of course, what was in EEG, had to match what was in MEG, such that people would know what to store and what not to store. And so, for example, sampling frequency is essential. Units are always required. What kind of electrode type, whether it's EEG, ECOG, stereo EEG, EKG, required. So there was there's a list of this that's in the BID state, in the brain imaging data structure of these kind of issues. Another discussion that we had with the community was whether electrode positions that we record with intracranial EEG, were those raw data, and should those be stored in BIDs? Or should those be derived data that are only going to be stored later in derivatives? And so, that was a discussion, because, for example, if you have data off the scanner, for example, in a NIFTY file, the DICOM file is the source data. Our electrode positions, how you get those of the clinical position, is from a CT, an MRI, or from an X-ray. You combine those, you integrate it. But then we decided that electrode positions and the locations, the coordinates of the electrodes are essential to interpret the data, and these should be stored in raw data. And the reconstruction step that goes before that is where you get the pictures from the neurologist, the electrode assignment table, etc., is a type of reconstruction. So that's why the electrodes, for example, are in the raw data and not in the derived data. And then you have, of course, start preparing examples for your extension. And what we did was a lot of comparison and matching and close collaboration with the MEG and the EEG extensions, which was absolutely fantastic. And so this is some of the content of what's in there right now. And you can see that, for example, here you have a data string. The JSON file shows some of these things that I've mentioned, such as the sampling frequency, task description, manufacturer. And then you can see, for example, for one of the channels, you can see, for example, there's a channel, the type is ECOG, unit is in millivolts here, low frequency, low cutoff, high cutoff, a notch filter, whether that's applied or not. And if you have all this information, you can plot the trace of the data with the proper units and the proper labeling in terms of time, etc. Your events then allow you to know at which time different events where different visual stimuli were presented. The electrodes, which are the hardware contacts, have a name that should match the channels and have XYZ coordinates that should be interpretable with respect to an image that is stored in the intended for field in the coordinate system. When you have this information, you can start making brain renderings with electrodes, for example. And then there's additional information that you can, for example, store, such as an X-ray or an operative photo that has the same label as the rest of this data set. And then there is the review and inclusion stage where things get reviewed by the BITS moderators, and then it can be included and approved in BITS. But there's this whole stream of things up to the, the BITS examples, etc. So after the Google Doc, where you have defined all these things, you can do a GitHub pull request where these things are translated into the proper BITS formatting. Examples have to be uploaded to the BITS examples page. The BITS starter kit has templates for storing all these metadata. You can validate your data. The BITS validator has to be updated such that the validator recognizes your data type and won't give an error. And then there's a review and inclusion. And so this is the workflow that we went through. And the interesting thing was that at every stage, whenever we updated, for example, the BITS validator, we had to go back and make sure that it matches the content of our data. When we were doing the GitHub pull request, we noticed that there were some things that were not the same between EEG and MEG and IEEG. So we had to go back and fix those. We had to make sure that the BITS examples were all up to date. And so whenever we found something in the validator, we had to go back and make sure that the BITS examples were all BITS approved. So this final integration was really a lot of work to get everything to match, be BITS approved. And there are still things, if you still have a data set and you find something little, please make a note on one of the issues in the GitHub page such that it can be improved. This is a community effort. Just to show you how this, now that we've extended the brain imaging data structure, how we can implement that in a clinical setting. I've been collaborating with people from the UMC Utrecht, and this is Doreen van Blois is one of the PhD students who's been working on this. And what they wanted to do is they wanted to use the brain imaging data structure to store all their clinical IEEG data that they use for studies on epilepsy. And those were both recorded during these long-term monitoring periods, as well as during interoperatively. And so you can see here that their source data was a set of clinical information that they had in a clinical database, IEEG data that they had in a clinical IEEG monitoring system. And then they had operative pictures that had the electrode positions. And so what's outlined in this paper is that there's many steps, and what those steps were to get these data properly structured in BIDS. basically how to get the annotations in there with the onsets of different phenomena, how to make sure that the data, that the data are, when you move from here, from the source data into the raw data, that's the step where de-identification happens. And that's a very important thing also if you think about the European, the GDPR rules. You have to make sure when you do the de-identification, this is all source data that can contain clinical information. This is all raw data that should be de-identified. So this paper outlines this workflow that they had in mind for not only storing long-term EEG data, but also storing these acute recordings where you may reposition the grid, do a recording, reposition, do a recording, etc. So how did they use the session structure that is available in BIDS to record these different situations? For example, situation 1, situation 2. It was all possible within the BID structure to record these clinical data with the accompanying pictures that were taken in the OR to get the electrode placement. For example, the events were stored, the channels were stored, etc. So this was quite a big effort to make sure that their pipeline and the way in which it was stored was set up correctly. And so these data and the paper with the workflow is shared online with 2 separate data sets that give an example of how you can implement, for example, the intracranial EEG BIDS in a clinical setting. And they have there's 2 data sets on Open Neuro, one that has the intraoperative examples and one for the long-term intracranial EEG data sets, where you can see an example for how this was implemented clinically in a larger setting. The examples that we have for research are already shared on the BIDS examples. So as an outline, I've shown you kind of why we were needing to have something like BIDS, why we needed to have that. I've shown you the BIDS folder structure and the metadata and some BIDS extensions and implementations. And then here are some links with the information that you can find online. This is the BIDS webpage where you can find all the information and links to everything. The BIDS specification that can be browsed for all the details of how to store your data in BIDS. A long list of examples with many MRI examples, MEG, EEG, all the intracranial EEG, all the accepted formats. I have an example on the BIDS example page. BIDS apps allow you to process these data automatically with a Docker container. And there's in this website, you can find how to basically build those Docker containers. The validator, such as you can use how to validate your data, is available online. And I would like to point also to the BIDS starter kit, where there's a wiki page that has templates and very straightforward information of how do I write and read a JSON file or a TSV file in Python, in R, or in MATLAB. Or where can I find a template of what my data should look like? What should my JSON file look like? What are the required fields, etc. So that you don't have to start typing from scratch when you go to the specification, but you can just have a template for EEG, MEG, intracranial EEG especially. For MRI, there are already a lot of templates available and standard procedures for automatically go from the DICOM files into the MRI extraction. Those tools are all highlighted in some of the BIDS apps, and this website also links some of the tools for MRI. But for intracranial EEG, as you saw before, that was not as straightforward, so we had to create those templates. So I would like to, thank my lab for all their efforts of putting all the data that we have in the lab into BIDS. This is a big effort where all the data that we're recording now. Gabriela Ojeda is the main person who is helping with that. And you've met Tal and Harvey, who are here at the NeuroHackademy this week. And also the IEG BIDS working group that made sure that actually BIDS works for all these different structures and that we can actually use it now in so many different settings. So I'm happy to answer any questions. Thank you.
Thanks, Dora. That was a really fantastic introduction into the world of BIDS and the specifics of all these details. It's really great. Let's open this up if people have questions.
Well, I have a question while people think here. Hey, Dora, thanks for the talk. This was really great. I'm wondering, you hinted a little bit at this in the talk, but I'm wondering if you could talk just a little bit about what you think are like the biggest problems to be solved in BIDS right now. And if, for example, I were a student at like a hackathon and I wanted to work on a BIDS-like project, do you have any thoughts about what kind of projects would be fall into that category?
Yes, yeah. So that's so that's one of the things that I wanted to highlight, the extensions. So the BIDS extensions really need help. And for example, we are now getting to the stage with the with all these different tools where we want to, for example, work on the. Let me just go back to this this particular slide right here. Oh, here, right here. For example, there's been several projects, I think, that highlight the connectivity data scheme. This is something that is definitely going to be necessary in the future, where we share connectomes between different labs in a standardized way. So if you're a student and you want to start contributing to BIDS, what I would do is I would click on this BIDS extension proposal. It opens the Google Doc and read through it. And if you have some suggestions, these Google Docs are the perfect stage where these contributions are heavily acknowledged. So if you want to make a contribution and you want to store your data or make sure that your derived data are BIDS compatible and your tool works with BIDS data, I would start to see whether you can store a connectome in the BIDS connectivity data schema. So when you read through it, make suggestions and say, like, this works for my data, this doesn't work for my data. And these, the nice thing is that in these Google Docs stage, all the contributions are heavily acknowledged. So at the bottom of these Google Docs, at the bottom, they generally have a section that says contributions, where you can basically, if you make a significant number of contributions, it's fair to add your name and someone will review it and say, like, okay, you read through it, made suggestions and made additions, and someone will approve that contribution. And that's what we also saw in our BIDS extension, is that some of these contributions, we asked for them. Like, we posted them on several social media outlets. So this is the current version of our IEG BIDS extension. Please make contributions to them. So these are several things. So if you work with, for example, electrophysiology derivatives, please post there, because we are actively monitoring those inputs and see what are the most needed. What's the most needed derivative structure for electrophysiology derivatives? For example, filtering, re-referencing, those types of derived data, adding in other annotations, for example. And these are just 4 examples. There's a list of 20 examples. So go through the extension proposals, click on the link and go through them. The other thing that you can definitely do if you're starting and you have questions, getting your data in BIDS, if you go to the BIDS starter kit and you don't find some information, you can actually change the wiki and make suggestions. So please do that, because that's the kind of input we need, is from people who are just starting out, trying to get their data in BIDS and are getting stuck somewhere or find errors. And all these contributions are acknowledged in either pull requests or in the BIDS structure in general.
Cool, thanks. And I just want to mention that we have a hack team working on connectivity data, and I'm guessing that they have probably already realized that your example about connectivity matrices is relevant. So thanks.
Okay, I see your hand is up.
Yes, thank you very much, Dora, for promoting BIDS. And I'm actually a user of BIDS, and I just want to say that it helps a lot of our facilitating our communication with clinical collaborators to find what the data is and how it is organized. And my question to you is I'm not sure if the BIDS community are aware of the anonymization pipeline that is usually run at clinical scanners when researchers get the anonymized data and specifically in DICOM format, sometimes there's some metadata missing there, which complicate running bits. So there are actually some data that is not passing the bits pipeline. And this is actually, we want to run bits, but we have to fix something earlier because of the anonymizations in the clinical setting. So do you or are the bits community aware of this problem and maybe can be addressed within the bits framework?
So whenever you, I think this is a great question, because this is kind of the, you run into an issue with your, I guess, the DCM to BIDS conversion. So I'm not aware of that issue, of course, because in our case, we work mostly with intracranial EEG data and don't use this particular pipeline. But if you run into these issues, these are active, these tools are actively being developed. So I would generally highlight that in make an issue, create an issue on their GitHub page, and that generally people will generally respond really well to that. Just say like, this is, if you can describe your issue really well. I've had good success with that. The other way where you can get in touch with some of the developers on these things is on the on some of the forums that are highlighted. There are several mailing lists where you can post questions that are readily addressed and picked up by some of the BIDS maintainers. So I can also recommend that